Look at the adult human body. Beneath the skin, there is a highly ordered, perfectly stacked column of vertebrae, flanked by symmetrical, segmentally arranged groups of skeletal muscle. To see where that structural order originates, we have to rewind to the 3rd week of embryonic development. At this stage, the embryo lacks any resemblance to a human. It is essentially a flat, featureless disc of rapidly dividing cells. Only a few central structures anchor this disc. Along the midline, we find the neural plate, which will become the central nervous system. Right beneath it runs a supportive cellular rod called the notochord. Flanking that central axis is a thick band of tissue called the paraxial mesoderm. This specific column of cells is the raw material that will solve the embryo's problem of structural order. If this raw paraxial mesoderm remained a continuous, solid sheet, the human body would lack its defining bilateral symmetry and segmented framework. The embryo needs a mechanism to slice this continuous sheet into distinct, workable parts. This transverse section shows how intraembryonic mesoderm divides into 3 zones. Nearest the neural groove is the paraxial mesoderm, followed by the intermediate, thinning into lateral plate mesoderm. Progressing tailward, this paraxial column organizes into paired concentric whorls called somatomeres. These paired somatomeres form right next to the developing neural tube, tightly linking the structural grid of the body to the central nervous system from the very beginning. Slicing a continuous tissue sheet into these preliminary somatomeres is the mandatory first step to converting a flat disc into distinct anatomical building blocks. These transverse sections show the next critical transformation. Mesenchymal cells of the somatomeres undergo epithelialization, packing tightly into paired cuboidal blocks adjacent to the neural tube called somites. As sharp boundaries form, a central cavity appears, the somaticule, marking the definitive somite. This animated timeline tracks their rapid appearance, running on a strict biological clock. The first pair emerges around day 20. New somites appear continuously, reaching 15 pairs by day 25 and peaking at 44 pairs by week 5, completing this segmented axis. Because they appear with such predictable periodicity, these temporary blocks act as an internal clock. The precise timing of their emergence dictates the exact schedule for all subsequent musculoskeletal development. A somite is not a permanent fixture. Once formed, it serves as a temporary staging ground before deliberately deconstructing itself. This diagram tracks that deconstruction. In panels D and E, cells in the ventromedial wall lose their epithelial borders and migrate inward, surrounding the neural tube and notoport. This forms the sclerotome, the direct primordia that will ossify into the vertebral column and ribs. Meanwhile, the cells remaining in the upper dorsal portion of a somite take a different path, forming a layer called the dermomyotome. The dermomyotome further splits into 2 groups. The dermatome remains in place to form the dermis of the skin on the back, while the myotome generates myoblasts, muscle precursor cells that migrate far out into the developing body wall and limbs to form our skeletal muscle. A cell's physical position within that original epithelial block dictates exactly what it will become, whether moving inward to build the body's bony armor or outward to build the muscular engine. Since all of these cells start out physically identical within the same somite, they require specific instructions to know exactly when and where to migrate. This diagram maps the chemical signals. At the bottom, the notochord and neural tube floor secrete sonic hedgehog and noggin, instructing the ventral cells to become sclerotome. Simultaneously, WNT proteins from the dorsal roof direct upper cells to form the dermomyotome. Further out, a balancing act occurs between the inhibiting protein BMP4 from the lateral plate mesoderm and activating signals from WNT and SHH, which finalizes the exact physical boundaries for the migrating muscle precursors. The complex physical architecture of the human body is actually a downstream manifestation of these invisible, highly regulated chemical gradients. By the end of the embryonic period, the distinct blocks of the somites have completely vanished, yet their legacy permanently forms our entire axial skeleton, the dermis of our back, and our skeletal musculature. Because the adult spine is built from this precise embryonic sequence, any stutter in the segmentation clock or misfire in the molecular signaling carries severe, permanent physical consequences. As this split screen shows, if an embryonic somite fails to segment properly, Or if the sclerotome forms asymmetrically on one side of the neural tube, the resulting adult vertebra will only develop in half. This direct mechanical failure causes conditions like congenital scoliosis and hemivertebra. In Klippel-Feil syndrome, patients are born with fused cervical vertebrae. This happens when the normal process of resegmentation, where halves of adjacent sclerotomes fuse to form a single vertebra, fails to separate properly in the early embryonic neck. Somites exist for only a brief window during the 3rd and 4th weeks of development, yet their highly regulated formation and deliberate deconstruction set the structural blueprint for human form, dictating our physical integrity for an entire lifetime.